Hello again. Here we are at the human respiratory system. So, in this video, we're going to look at several different areas. We look at firstly respiration, then we look at the structure of the human respiratory system, and then the final part that we look at is gas exchange. Now, I always think one of the first things we should look at is these little questions that people always get wrong. There's a lot of keywords here and a lot of things which sound the same and a lot of people always get mixed up. So firstly, we've got breathing, we've got respiration, and we've got gas exchange. Okay, so let's have a look at them. What's the difference between breathing and respiration? Because I guarantee if you just go and ask somebody who doesn't really know what's the difference between breathing and respiration, they'll be like, are they not the same thing? But actually they're not. So breathing is inhaling and exhaling. So basically, whenever we breathe in air and then breathe out air, and more specifically, what takes place during breathing is oxygen enters the lungs and goes into the blood, and carbon dioxide leaves, and that is called gas exchange. Respiration, though, is something different. It is using the oxygen to release energy from our food. So, breathing is inhaling and exhaling. Respiration is using the oxygen that we breathe in to release energy from our food. And as well as that, it takes place in the cells all around our body, whereas breathing only takes place in the lungs. Now, the next one that people get confused about is where does gas exchange take place? Gas exchange takes place in the alveoli of the lungs. Now, those are the little air sacs that we're going to look at later on, and we'll look at how they're adapted for gas exchange. And then the next little part, where does respiration take place? It takes place in every cell in the body. Every cell has to be able to respire. If it doesn't respire, it will die after a couple of minutes, which is basically what happens in a heart attack. The cells in your heart can't get enough oxygen, so they can't respire, so they die. Now, first little section of this booklet is looking at cell respiration. Now, respiration is a series of chemical reactions in every cell in the body that releases energy from our food, and it needs oxygen. The reactions take place in special structures in the cell cytoplasm called mitochondria. Mitochondria. Now, this diagram shows a typical animal cell, and this is just to show the equation for respiration. Now, we should know that the equation for respiration is glucose plus oxygen, which gives us or releases carbon dioxide and water and energy. So, glucose and oxygen are the raw materials, carbon dioxide and water are the waste products, and the main point of respiration is to release energy. And we'll look at what that energy can be used for in a minute. But if we get back to this cell, so this cell gets glucose from our food, so probably from digesting carbohydrates. We also get oxygen from breathing. Those two things go into the cell, and then in this part, which is the cytoplasm, you will find the little mitochondria, and that's where respiration actually takes place, in the mitochondria. So the oxygen and the glucose have to diffuse into the mitochondria, they react together, and it releases carbon dioxide and water. Now, the carbon dioxide will travel to the lungs and get breathed out, and the water will also get breathed out, or it'll be removed in your pee, in your urine. And the final thing then is the energy that's produced. So this was the whole point of respiration. It's used for growth, repair, movement and heat. Okay, now we talked about the energy and what it's used for, but this just gives you a bit more detail about them. So the first one is active uptake or transport. Now, we did talk about this whenever we were looking at ecology and root hair cells, but active uptake is the movement of substances against a concentration gradient, and it requires energy. So just to give you a bit more detail on that, in diffusion, particles diffused or moved from an area of high concentration to low concentration, whereas active transport was the opposite. It was from low concentration to high. So you need energy for that process. Other processes that require energy is movement, which is pretty much self-explanatory. Growth, again self-explanatory. Reproduction. Now that can be to produce sperm and eggs to actually be able to reproduce, or it can be the energy needed to grow the offspring. And also, energy is needed to maintain a constant, it would be good if I could spell that, a constant body temperature. The next little part then is the word equation for aerobic respiration. So as we said, we get glucose from digesting our food, oxygen from breathing, it releases carbon dioxide, which we breathe out, water, which we can breathe out, we can also sweat it out, or we can get rid of it in your urine, and energy, which is what we came here for, the energy. Now, if we look at this, there's a balanced equation for this as well. Now, you don't have to do chemistry to understand this. You don't even really need to understand it. All you need to be able to do is rewrite this, or if they give you parts of it, fill in the blanks. So the glucose is C6H12O6 plus 6O2, which gives you 6CO2 plus 6H2O plus energy. Now, when you're revising this, it's important that anywhere I've used small numbers, you have to write them out as small numbers. Anywhere that I've used big numbers, they have to be in big numbers. And anywhere that I've used capital letters, you have to use capital letters as well. Now, the final wee part about respiration before we move on and look at anaerobic respiration is this is an exothermic reaction because energy is given out. Now, there's lots of reasons why this is important. The first one is we said that respiration is used to release energy, and it's also, because it's exothermic, it produces body heat. So the more respiration we carry out, the hotter we would get. So for example, when we exercise, we get very hot because our muscles are respiring more. If we go into very cold climates or cold weather, you'll need to respire more to maintain your body temperature. So that's why you might eat more during really, really cold weather. As well as that, if you had lots of bacteria growing in a big biodigester, it would get very, very hot and you would need to control the temperature. As well as that, in ecology, we talked about how decomposition or decomposing material might get very hot because of the bacteria respiring and releasing heat. So there's lots and lots of places that we've talked about this before. So this next little part then is about anaerobic respiration. Now this takes place when there is not enough oxygen available. Some cells, for example, muscles and organisms like yeast, can release a small amount of energy in food molecules using a series of reactions known as anaerobic respiration. So the key difference here is that aerobic respiration takes place with oxygen and anaerobic takes place without oxygen. Okay, now it is possible for muscles and other organisms to respire without oxygen. Usually, if we're talking about mammals or humans, it's generally only a very, very short period of time. And we'll look at some examples. Now, we're starting off here looking at yeast. So, yeast are one of the organisms that can respire anaerobically, which means that they respire without oxygen. So what they do is they take in glucose and they respire and produce alcohol, carbon dioxide, and energy. And this is pretty much how beer and wine is produced. Yeast is added to a container with glucose. The oxygen supply is cut off from them and they respire and produce alcohol. And there's actually a little experiment you can do to show anaerobic respiration taking place and also the product it produces. So basically what you do is you take a test tube, let's call this test tube A, and in that you put these two things. You put a glucose and a yeast solution, and on top of that you put a layer of oil. Now the layer of oil prevents oxygen from entering the solution. As well as that, before you add the yeast to the glucose, you make up a glucose solution and you boil it and cool it, and then you add the yeast to it. Now the reason why you boil it is to remove the oxygen, because I'm sure you've noticed whenever you boil something it bubbles, and that's just the oxygen leaving the mixture. You then cool it down so it doesn't kill the yeast, and then you add the yeast to it. You put it into the test tube, you put the oil on top of it, and you leave it for a little while. Now what you'll see eventually are little bubbles of gas, and the gas here is going to be carbon dioxide, because remember they're going to be respiring and they produce alcohol plus carbon dioxide. So the little bubbles that they produce are bubbles of carbon dioxide. It travels up through here, through the delivery tube, into the second boiling tube, and in here we have lime water. Now the lime water will turn from colourless to milky if carbon dioxide is present. So this little experiment shows us that when we take glucose and yeast and put them in anaerobic conditions with no oxygen, it will still respire, produce carbon dioxide, which turns the lime water milky. Now typical questions you'll get from this: the water used to prepare the sugar solution and yeast suspension was boiled prior to use. Why? Firstly, it's to remove all oxygen from the sugar and yeast mixture so that it ensures anaerobic conditions. So i.e. there is no oxygen present. Why is the layer of the liquid paraffin, or what it was in my diagram was oil, placed on top of the yeast or sugar solution? It was to prevent air entering the mixture, so there was no oxygen present. What results would you expect for this experiment? Explain them as fully as you can. Firstly, the lime water turns from, let's say, colourless to milky because the carbon dioxide is produced. Alcohol will be produced. Now, of course you could test for that, but another way to test it would be smell it. And finally, the solution will be slightly warmer due to respiration taking place. because respiration is, well, it releases energy and it is exothermic. What would a suitable control for this experiment be? It would be the same apparatus, but the yeast would be replaced with water. Now, remember we've talked about controls before, and we said the point of a control is to prove that the living organism is causing the results that you see. So, in this experiment, the yeast is the living organism. So if you remove it, what should happen is that there will be no respiration taking place because there's nothing living. So the lime water will not change. So therefore, it proves that the yeast is causing the change in the lime water. Now, before we started anaerobic respiration, we said that yeast can respire anaerobically, as can human muscles. But we said it can't take place for very long, so it's only a short period of time. Now that's only if aerobic respiration doesn't meet their energy needs, so it basically can't release energy quick enough. This is likely to happen during strenuous exercise and lead to muscle soreness. So I can guarantee that everyone has felt this before. Now, if you actually pause it here and do this, raise your hand and make fist movements for as long as you possibly can. What happens? Like this. Hello. Hand up. Hand up nice and high. Fist. Go. And do this for as long as you possibly can. Now, what you'll notice pretty quick is that your hand starts to get sore. And if you do it for ages and ages, you might even get a cheeky little cramp in your arm. And the reason for this is the muscles in your hand run short of oxygen and start to respire anaerobically. So basically, the oxygen cannot get to your muscles fast enough, so they start to respire anaerobically, but they also start to get a little bit sore. Now the reason they get sore is because of this, which we can demonstrate with a cheeky little word equation. So we still use glucose, but this time there's no oxygen because it's anaerobic, and we get lactic acid building up in our muscles. But we also get energy release. So the energy is what we can use to move, but the lactic acid is what makes your muscles sore. And then this leads on to the next bit. Why can we not maintain a top speed sprint for more than a matter of seconds? So this is basically because during vigorous exercise or very strenuous exercise, the heart and lungs can't deliver oxygen to the muscles fast enough for aerobic respiration. So they respire anaerobically and produce lactic acid. Now that was a bit of a chunky pen, so there, that's better. So they respire anaerobically and they produce lactic acid, which gets sore and causes cramp. Now here's a thought. So if anaerobic respiration makes your muscles sore and it causes cramp because it produces lactic acid, why do animals even need it? Well, there's a few reasons. Firstly, to escape predators, so to stop them being eaten. It might also be if they're sprinting to catch prey. and then any reason why they might need to escape danger. So pretty much in an emergency situation where they just can't get oxygen to their muscles quick enough, that's when they need anaerobic respiration. Now, here's the equations we've got for anaerobic respiration. So remember, anaerobic respiration is when there is no oxygen involved. So, in yeast, we sort glucose and you get carbon dioxide plus alcohol plus energy. But in mammals, you get glucose plus carbon dioxide, which produces carbon dioxide plus lactate or lactic acid plus energy. Now, one of the big things that you're going to have to do at GCSE is compare aerobic with anaerobic respiration. So, you can work out some differences by comparing their equations. So if we look at the aerobic respiration equation, it is glucose plus oxygen, which produces carbon dioxide plus water plus energy. Now, we should say that there's lots of energy produced in aerobic respiration, whereas anaerobic respiration, we get glucose, which produces carbon dioxide plus lactate or lactic acid plus energy. But in this case, there's only little. Okay? Now, if we compare these are the things that we could talk about. So from the equation, we could get the substrate, oxygen present, carbon dioxide produced, and other products. So aerobic and anaerobic both use glucose. Aerobic needs oxygen, but anaerobic doesn't. Carbon dioxide is produced in aerobic, but it is only produced in yeast in anaerobic respiration. So we could say not in muscles. Other products that we'll get is water in aerobic, but in anaerobic, alcohol or lactic acid. And then the energy produced is loads in aerobic, but very little in anaerobic. The final little section that we're going to look at in this video then is factors affecting the respiration of yeast. Now, the two that we would mainly look at would be temperature and the type of sugar. Now, up until now, we've said that respiration needs glucose, but actually, different types of yeast can use different types of sugar. So, the first one that we're going to look at is temperature. Now, the aim of this experiment is to investigate the effect of temperature on the respiration of yeast. So this is the apparatus that you use. You use a water bath, and that's pretty much used, as it is always, to control the temperature, or to change the temperature in this case. You've got a thermometer to measure it. You've got water in the water bath, and within that you've got a boiling tube, and inside the boiling tube you've got an upside down test tube which contains glucose and yeast solution. You've also got a little gas bubble. Now here's your method. It's unlikely that you would need to learn this, but let's have a look at it anyway. So what you do is make a yeast solution with glucose with 100 centimeter cubed of boiled, cooled water in a beaker. Now remember, we need to boil it to remove oxygen and cool it so it doesn't kill the yeast. Then you leave it room temperature for 1 hour. That just gives it time for the yeast to absorb the glucose and to start respiring. You then fill a test tube with the mixture. Place the boiling tube upside down over a small test tube containing the yeast mixture and invert both tubes or turn upside down. Place the tubes in a water bath at 10 degrees C. So that is our first temperature. You measure the height of the bubble at the top of the small inner tube. So you start off by measuring the height of this bubble. Can you draw me a line, please? You measure the height of that. You repeat steps 2 to 6 at temperatures 20, 30, 40, 50. So altogether, we'll have measured 10, 20, 30, 40, 50. So we'll have 5 readings. And then after 30 minutes, measure the height of the bubble at the top of each of the inner tubes. So this is what we're expecting to measure. We start off with our boiling tube. We've then got our test tube upside down with a gas at the start. We measure that in millimeters. We then leave 10 minutes and we take it out of the water bath. We measure it, and this gas bubble at the end, measure it in centimeters or measure it in millimeters because that's what we said the first time, and it should have got bigger. Now, the reason it will have got bigger is because respiration will have taken place and it will have produced carbon dioxide. So this gas up here is carbon dioxide, and by leaving it to respire for 10 minutes in the water bath, more carbon dioxide would have been produced, so the gas bubble should have got bigger. Now, this is sort of what it would look like in our table of results. So this was the height of bubble at the start, this was it after 30 minutes, and this was the change in height. Now from these results here, we can clearly see that at low temperatures, there's a very small change in height. The change gets bigger and bigger up to 35, but then above 35, the change in height starts to decrease again. Now you would then draw a line graph of your results. So at the bottom, you put temperature, and at the side, on the y-axis, on the x you put temperature, on the y you put the change in height, and these are the sort of results that you would see. So we can see the largest change happens at 35 degrees. There's less of a change in height at low temperatures and at high temperatures. Now, in conclusion for this experiment, there's a few questions that we need to answer. Why was the glucose boiled and cooled before the yeast was added? What type of respiration is the yeast using mostly? Which temperature was the fastest? And explain the results at 10 degrees and 50 degrees C. So why was the glucose boiled and cooled before the yeast was added? It had to be boiled to ensure that there was no other microbes contaminating the yeast and to remove oxygen to make sure that it's anaerobic. It needed to be cooled to ensure that the yeast were not killed by the high temperature. What type of respiration is the yeast using mostly? It's anaerobic because there's little oxygen that can get to the yeast at the top of the test tube, and the oxygen was removed by boiling. The yeast in the middle or the bottom of the test tube will therefore not receive oxygen. Which temperature was the fastest? It was 30 to 40 degrees C, and that's because that was the highest change, or in fact instead of highest, it was the greatest change in height. Explain the results at 10 degrees and 50 degrees C. At 10 degrees C, the molecules have less kinetic energy, so the rate of reaction was slower. As well as that, and this is important, respiration gets faster as the temperature increases. So at low temperatures, the rate is slow because the molecules have less kinetic energy, so the rate of reaction is slow. At 50 degrees C, the enzymes involved in respiration are denatured at that temperature, so the rate of the respiration decreases. Now, what does denatured mean? You might remember from enzymes, that is when the active site changes shape and the substrate no longer fits. Now you might be thinking, well, how the hell do enzymes come into this, right? But remember, yeast are living things. They contain enzymes. And if they get too hot, those enzymes will get denatured. And because respiration relies on different enzymes, if it gets too hot, respiration will stop. So the second experiment that we can look at to investigate respiration is the type of sugar. Now, in this, we're going to use 5 different types of sugar. You'll notice that they all end in OSE, which is typical of sugars. And the thing about this yeast is that it can use any of these sugars, but we're going to find out which one it can use best. So in this experiment, we're going to do a very, very similar experiment where we have the boiling tube upside down with the yeast and the sugar. And this time all we need to do is set it up a few times and change the type of sugar. So what we do is we measure the height of bubble at the start and height of bubble after 30 minutes and measure the change in height. Now, if we just look at the method really quick. So again, we make up a yeast and glucose solution with boiled cooled water. We leave it room temperature for 1 hour, fill a small test tube, invert them, place the tubes in the water bath at 30 degrees C this time, measure the height of the bubble in the small inner tube, and repeat with yeast mixtures containing the other sugars. Record your results in the table and calculate the change in height, and then draw a bar graph this time. Now, I suppose the main differences are that we're only using one temperature, so we need to keep that the same to make sure it's a fair test, but we are changing these, so these are going to be our, or this is going to be our independent variable. This is going to be a controlled variable. Now, if we look at sample results here, we can see that with glucose, there was a large change in height. With sucrose, it was very small. We also had a large change with fructose and a smaller one, well, a change, but very similar with lactose and maltose. So we take our results, we plot them in a bar graph. This time we put the different types of sugar on the x-axis and the change in height on the y-axis. So, in conclusion then, fructose was the best sugar that yeast could use for respiration, glucose was second, followed by maltose, then lactose, and sucrose was the hardest one for it to respire. Now, this might just mean that for sucrose, lactose or maltose, it may have to carry out additional reactions before it's able to respire it. So for example, glucose and fructose, it can just absorb it, respire it straight away, whereas the other ones, it might actually have to go through a slightly different reaction to be able to respire it and release the energy. Or it might just be that sucrose, lactose and maltose just don't produce as much energy during anaerobic respiration. So therefore, if they don't produce as much energy, they will not produce as much carbon dioxide either. And that's the end of the first section of this topic. Thank you. Goodbye.